Okay, so welcome back. We are now going over mitosis. There are 5 official steps to mitosis. Well, interphase, which is the first one, isn't necessarily a step of mitosis. It comes before mitosis. So the next 4 steps would be prophase, metaphase, anaphase, and telophase. There's also tokenous, but that isn't a part of the mitosis. It comes right after telophase, where the two cells split. So now, if we look on interphase is where the cell is engaged in metabolic activity and performing its duty as part of a tissue. The DNA duplicates during interphase to prepare for mitosis, or the next 4 phases that lead up to and include nuclear division, like I was talking about. Chromosomes are not clearly discerned in the nucleus, although a dark spot called the nucleus may be visible. That's how you know it's interphase. Interphase again. Interphase is the longest stage of mitosis. The cell carries out various functions in the organism, and the cell doubles everything in the cytoplasm. Then, the prophase. So chromatids in the nucleus begin to condense and become visible in the light microscope as chromosomes. The nuclear membrane dissolves, marking the beginning of the prometaphase. Proteins attach to the centromeres, creating the kind torches. The microblues attach at the kind torches, and the chromosomes begin moving. So they look like bow ties, apparently. So the centrioles lie in a region called the centrosome. The centrosome helps to organize the spindle, or these red things, and a fan-like microtubule structure that helps separate the chromosomes. Metaphase. Spindle fibers align the chromosomes along the middle of the nucleus. This line is referred to as the metaphase plate. This organization helps to ensure that in the next phase, when the chromosomes are separated, each new nucleus will receive 1 copy of each chromosome. So they line up in the middle like line dancers, like, the river dancers, never mind. Anyway, so metaphase is the 3rd phase of mitosis. And the chromosomes line up across, so like we already talked about this. And the centromere connects to the poles of the spindle, which are the 2 red marshmallows, apparently. Anaphase. This is the paired chromosomes separate at the kinotorches and move to the opposite sides of the cell. Motion results from a combination of kinotorcho movement along the spindle microtubules and through the physical interaction of polar microtubules. I don't know how to say that, but that's apparently how we're saying it. So, they're pulling apart. It's going to the end. Now, this is also it. So, anaphase, chromosomes break at the centromeres, and the sister chromatids move to the opposite side of the cells. These are the sister chromatids there. Now, we're talking about telophase, which is the last stage, officially the last stage of mitosis, or the end of PMAT, which is, PMAT. Anyway, telophase, the new membranes form around the daughter nuclei, nuclei is a plural for nucleus. While the chromosomes disperse and are no longer visible under the light of the microscope, Mitosis, or the patrolling of the cell, may also begin during this stage. This is also it. This is a nice little doodle someone made. There, that's a pretty good doodle. Anyway, this isn't the step of what you would call mitosis, but it is the last step to separate the cells. So, cytosis, as mitosis ends with the cell nucleus being divided, cytosis begins. During cytosis, the cytoplasm of the cell is divided in half, and the cell membrane grows to enclose each cell, forming two separate cells as a result. The end result of mitosis and cytosis is two genetically identical cells where only one cell existed before. So this is what mitosis is. And so this is how you get new cells. Yeah, there we go. So goodbye. I think that is all for today. Please like and subscribe, and comment me if you think I should do something better. Please give me suggestions. Okay, thank you, and that's all. Bye.